Yeah, well, then we are ready. And I see there's a lot of audience that sign up to the presentation. First of all, I would like to welcome everybody, especially Kåre Enkilde from Amniotics. This is a company introduction to Amniotics. And secondary, I would like to welcome all the participants in this HC Andersen event. Amniotics entered the stock exchange the first trading day, the 6th of July this year. The current market cap is 192 million Swedish kroner, and Amniotics is specialized within stem cell therapies with a very interesting business model and a couple of promising projects. So I would like to hand over the presentation to the CEO, Kåre Enkilde.
Thank you, Klaus, and thank you for this introduction. You can say this will be a bit of a complicated understanding of the products that Amniotics work with, so I'm very open for getting into discussions and discussing our whole process moving forward. If we start out, maybe it would be important to understand what amniotics means. You could say in Danish, it's or amniotic fluid. In Danish, it's fostervand. In Swedish, it's fostervatten. So basically, you could say amniotic fluid, which is our starting material, is the fluid that surrounds the child or the neonate inside the room. I think that's a good beginning to start off with for people to understand. If we take it, Amniotic at a glance, we were founded in 2015 in southern Sweden, located at the old Astra sites here in Lund. And we are based on research from Lund University Stem Cell Center as well as the Lund Hospital. We have 3 founders, so we have both clinicians and stem cell researchers as founding fathers. We've built a full GMP process facility enabling us to produce our products all the way to clinical trials, but even all the way forward to market. I think that's an important understanding as well. As Klaus already alluded to, we were listed here on July 6th, and we have a pipeline targeting several different markets. What we do and what our focus is to develop and manufacture innovative stem cell therapeutics to treat severe life-threatening diseases where we see a lack of effective treatments. Our stem cells, as mentioned earlier, is based on the amniotic fluid or taking from the amniotic fluid, where we see a unique richness in the high-quality stem cells. And we are able, you could say, because if you think of the fluid, it's kind of a reservoir or deposit for the cells from the neonate, from the child, that will be collected into that fluid, and we can then take that fluid and select the cells from that. The whole process of working with that is taking the fluid from planned C-section. I'll drill a little more into that later on. The business model is, you could say, type of the customary biotech model, high risk, high gain. But what's also important to understand here is that Amniotics has a technology platform that we can build upon, and we can continue expanding our processes all the way forward. But also understanding that we actually are very uniquely positioned because we both have a medical device that we use to get or harvest the amniotic fluid, source the amniotic fluid, all the way to manufacturing and production, and then even to always forward to fill and finish, and later on to the market or clinical trials. That opens up for secondary business opportunities. So you can say if we move away from our clinical products, we can either do, you could say, production of other products for other companies or hospitals and academias. What we primarily work with right now is we work with respiratory indication, neurodegenerative diseases, nephrology, and dermatology. So we already have 4 big areas that we focus on. As mentioned earlier, it's focusing on these cells selected from the amniotic fluid. This is, you could say, to understand also that a stem cell product is very different from what you would see for small molecule and antibody, meaning that being in the clinical trials and having data there, you're already there opening up for a broad setup of different treatments. Because this is a cell therapy, it's more, you could say, broad, applicable for many different disease areas and not just a single one. So we're certainly not a one-trick pony, neither in the indication nor in the product side. a little dwelling into our technology platform, and maybe I should start out with explaining a bit about what is stem cells, because that is, you could say, maybe a bit of difficulty sometimes to understand. And you could say, to start off with, stem cells are understandable as a clone that can become more clones. So you have what you can call expansion. So a single one can be millions of cells. But even that single clone will be able to develop into specialized cells, each of them. So you can think of stem cells as the wiring or the starting material for all the cells in our body. And from there, you even have different types of stem cells. What amniotics primarily work with is what's called mesenchymal stem cells, but also we have something on the induced pluripotent stem cells. We'll drill a little onto that later on in this presentation. But to start off with here, I think it's important to understand that these cells are anti-inflammatory or inflammatory modulatory, and they are anti-fibrotic, meaning they can remove some of the scarring you sometimes see that affects the tissue following an insult, and that insult can be an inflammation, or it can be a direct trauma to that tissue or organ. Where we see our product, and you can also readily here see that we are able to source the amniotic fluid from the planned C-sections or cesarean sections. Our product is used, you could say, when the obstetrician has used or made the first incisions, and the amniotic membrane, in Danish fosterhinden, in the same Swedish. Our device is used to penetrate the amniotic membrane, and then the fluid runs into our device, a process that takes roughly 90 seconds. So not an extensive, you could say, setup, but a rather quick setup, enabling us to get that fluid and then use that for developing our therapies. So it's kind of, you can think of life treatments to life. So we are taking something that's very important and even saving lives with that. Also important to understand here is that a single donation of amniotic fluid will run enough for treatment thousands of patients. Roughly, we're thinking about 6,000 patients, all depending on indication and dose for that indication. As mentioned earlier, we have a full manufacturing facility. We actually was approved by the Swedish MPA Läkemedelsverket here in late 2020. So we've built a full production facility with QC labs and all of that inside that. So we're able to manufacturing our product ourselves, enabling us, you could say, to go all the way to market and also all the way to clinical trials, enabling us to be right in time and selecting the products we want to produce at the right time. And at the same time, enabling us to do contract manufacturing and developments for other companies that lack this competence. This is an overview of our process, and as you can see here, I alluded to it earlier, we both have a CE marked and patented device to sourcing of the amniotic fluid. Then that fluid is transported to our facility where we filter it and select it and expand these cells. So you could say one cell become billions of cells. And then these cells are ready to fill and finish, all ready for clinical trials and later on for the market. That then opens up for us being able to both continue developing our own clinical trials, but also out licensing at different stages of developments. Our pipeline is right now primarily focused on the pulmostem product we have for respiratory indications. We are looking at COVID-19 ARDS that we are targeting first half next year. Then we're following up with idiopathic pulmonary fibrosis or lung fibrosis, a chronic disease that is very difficult to treat, and there's no treatment presently that's really very efficacious. And then we're also looking at lung transplantation, again, an indication that's really in need of good treatment, because several of these patients that get a lung transplantation gets what's called primary graft dysfunction that can later on lead to that transplant needed either death or that retransplantation. And then we start to broaden our pipeline into the neurodegenerative space, into the skin, the kidney, and then lastly, what we call the blood space. We are able to produce what's called hematopoietic stem cells, the same kind of product you use for bone marrow transplantation. This is an overview of the market because, of course, it's important to understand how does a market for cell therapy look like. It's a very nascent market, as you can also see, because this is, you could say, early stage. Despite clinical trials with this type of product has been, you could say, done for quite some years, there's still a lack of moving all the way to clinical trials and all the way through to market. So there's a need to reach that, but that very much looks like what it will become in a few years from now. So a very rapid increase as we forecast from global data. This is for the 8 major markets, just a small, you could say, extra understanding. Also an understanding of how does these markets that we target look like. As you can see here, we have the CNS space, the neurodegenerative space, we have the kidney, the respiratory, and the derma space. All of them are very much looking to grow kind of rapidly in a few years' time, especially for dermatology and neurodegenerative. But also kidney looks to be increasing quite rapidly because some of the type 2 diabetes, some of the cancer treatments with cisplatin and so forth end up in destruction or attacking the kidney and decreasing that efficacy and that needs treatment. So goes for many of these indications, and many of them are due to, you could say, an aging body, an aging population. So that's why you see a lot of these indications going upwards in treatments or need for treatments. Since we're working with cells, you could say we also focus on orphan indication or small indication because this is where we really see a good niche for this type of products. It also where we see that there really is a lack of treatments in many of these indications, some of the small indications. Also understanding from a biotech perspective, this is also where you don't need as big clinical trials. So enabling us to move rather rapidly and understanding also that when you're on the market and later on, you can expand further on beyond that. So when you're on market and have an approved product, that then can later on be expanded into other indications. You could say this slide kind of speaks to the earlier slide on how we view the forecasted growth of cell therapy. You can see here, I'm not dwell into details, but just you can see how it increases in the sizes of these deals, growing from 2017 when it's rather small size deals and all the way up till just last year, which was rather big. Also, of course, important to understand how we work with our IP and how we expand our IP. This is, you could say, focusing on taking each step of the way and making sure that we have a good structure of our patent around that, both on the different products, on the process we work with, and even on the indication. So we're really growing a very tight and strong patent portfolio as we move forward, which we believe to be very, very important. Then a small appetizer on the induced pluripotent stem cell therapy. This is, you could say, something that may become a big thing 15 years from now or something like that, because this is also a very important area. As you can see here, Amniotics has some core patents around how to use this product to produce what's called hematopoietic stem cells that can later on be used for bone marrow transplantation or even moving into oncology and cancer treatments. Our value drivers, as we look at it, as we alluded to earlier, if we look at it, the global stem cell therapy market is really nascent presently in the regulated space, but it's really looking to grow very rapidly. So very quick increase in this space. Amniotics, as mentioned earlier, also controls the full manufacturing process value chain. And if one looks at it, that opens up for higher pieces of the pie if we look at licensing deals and so forth. Also, we are working with what's called tissue relevant because we select the cells based on their relevance, focus towards the tissues. So we're working with tissue relevant and tissue specific neonatal mesenchymal stem cells that are tailored for the different treatments. So you could say likes treat likes, liver treats liver, or kidney treats liver. I think that's an important differentiation mark also. We have a broad pipeline that is really targeting attractive specialty disease market. I showed them earlier on. All of them pretty much grow quite rapidly, especially if we look at derma and neurodegenerative. We have the ability to license to pharma companies at different stages of development and offer also manufacturing services to others, which have a potential to create higher value in orphan drugs. As Klaus also already alluded to, We had a listing on Nasdaq First North this summer, where we raised 60 million SEK, and really growing our you could say, ability to grow, but also in the clinical space.
So, Kåre, can I just very short ask you a question regarding the 60 million that you actually got from entering the stock exchange? So these 16 million, I know you have a plan for how you're going to use those. You even have milestones on the book. Could you elaborate a little on how you plan to use these 60 million?
Certainly. You could say what we're using that for is mainly clinical development. For people who have worked in biotech and pharma space will know that the clinical stages are so often the areas where it's most costly to develop your product. So one really needs to have the foundation to work on, and from there you need to grow into clinical stage. And I think that's where we are. That's why we focus a lot of our attention to, and that's where most of the 60 million will be spent in the clinical space, developing the core documents that you need, seeking where one should go for the clinical trials, what nations or countries, and so forth.
Good, thanks. And just to the audience, if you have questions for Kåre, just post them in the chat down at the right, and then I hand over the mic again to Kåre.
Thanks, Klaus. And I think this is just, you could say, the end slide. This is just to show that we have a very competent, experienced management team. We have myself with a background from some of the major players in Denmark. Also Johnny Humajoya with a strong background, also both from medtech, but also biotech and biopharma. We have Helle Størum with a background from both Lundbeck, but also Pharmacosmos and Zealand Pharma, having helped them in building their business. And even Jan Tals, a very strong academics with a strong background from Germany, Sweden, and Denmark, so really building around that area with a strong academic understanding and also with a biotech experience. So that would be the last one, and I'm very open for any questions that haven't come yet.
Good, thanks a lot, Kåre. Well, I can pose the first question then. You mentioned early on in the presentation, when you extract amniotic fluid from one patient, it lasts for 6,000 patients. So just to understand this right, so it's not cells to cure the patient when the patient get born and then grow up. You can actually use this for other patients as well, and for, of course, different kind of diseases.
Yeah. I think you could say, to start off with, this tends to be a little scientific, but you could say there's two words that needs to be understood here. One is what you call autologous treatment, where it's the same patient's cells that is used to treat that same patients. And then you have what you call allogenic treatments, where it's cells from someone else that are used to treat someone else. So, and what we work with is allogenic treatments that gives, you could say, scale, enabling us to have a single donation that can be treated up to 6,000 or more or less, of course, all depending on dose. It's difficult to say exactly that, but that is our initial, you could say, calculation when we think about the dose that will be needed. Good.
Then there's one question here regarding the competitive landscape. You mentioned that in your presentation. Sorry, but could you repeat that again, Kåre?
Certainly. You could say the competitive landscape, and that of course also depends on how one chooses to view that. One can choose to say, as these products are inflammatory or anti-inflammatory, or inflammatory modulatory and anti-fibrotic, one could start thinking about what is the landscape there. And there I think it's a rather big landscape, but also to understand that many of the competitors in that space will be based on small molecules or antibodies. Both of these type of products are focused on a single target, while a cell is a more, you could say, able to be used in different settings and will respond to the environment it's in. it's a living product, hence it's able to respond to that specific environment, being it inflammatory, be it fibrotic, or something else. So it's a kind of different setup if you think of it that way. Then if we think about our peers in this space, I think we have a differentiation. Our starting material, I think, is very unique. I think the amniotic fluid is giving us some very unique characteristics because we have neonatal fluid or fluid with neonatal cells that enabling us to have some of these quick expansion of the cells, meaning they grow fast. Also enabling us to have this tissue-specific or tissue-relevant cells. I think that's also some very differentiation understanding. Because there are competitors, we also looked at on the previous slides when we saw the benchmarks. There are some competitors or peers in this space, but I think that's also very important to have that because that can also be kind of someone that is able to move forward and start getting that interaction with the regulators in the end trials and the end phase 3 trials and get that discussions going.
And then this uniqueness about the GMP, so you got the production in-house. If you compare that to the closest peers, is that normal that you build up your own production, or it's more normal that you actually source that out? And what kind of strength is that giving to you, both in terms of course, partnerships, as you mentioned, but also when you have to scale up? I could imagine that's a critical factor.
We certainly believe so. I would say I wouldn't say too many of our competitors have that capability of having a full GMP setup, and we also, you could say, we build it to be able to expand, so we can have several pipelines going at the same time in our GMP facility. So we are not, you could say, limited by space in our GMP facility. That will not be until several years from now.
Okay.
Having that GMP facility opens up also, you could say, both scale. It opens also up to collecting IP and know-how when you produce the products, constantly getting that and strengthening one's patent portfolio. It also opens up to understanding how one works with a GMP product. I think that's very important with a cell therapy because it's a more complicated process. If one looks at how small a small molecule is, it's small. An antibody is also quite small, and then if you look at a cell, it's much bigger. So it's a more complicated setup, it's a more complicated product. But knowing how to work with that also opens up to having scale, meaning that, and even positioning, because we can take this process and then, okay, we want to do the same in the US. Okay, so we can set up a production facility in the US if needed, or we can set one up in APAC, China, Japan, where we think there's a big need here. So that enables us to have that know-how, that knowledge of working and enabling us to do that. Because other companies, if you think of the biotech space, other companies have not necessarily that capabilities, and some that have been able or have burned actually because they didn't know exactly what to do.
Good. Thanks a lot. I don't know if there's further questions for Kåre. If not, I don't have further questions, Kåre. I just want to, first of all, thanks a lot for great presentation, Kåre, and thanks a lot for the audience to participate in this very interesting introduction to amniotics. Have a very nice day. Thank you.
Cheers.
Cheers.